All right, it's about 12:30 Eastern, so we'll go ahead and get started. My name is John Mann, and I am an assistant professor here at Michigan State University, and I host this webinar series called Innovations in Agriculture and Rural Development. And the series is sponsored by the North Central Regional Center for Rural Development. There are 4 centers in the U.S., and Michigan State houses the North Central region. So I want to welcome everybody to today's webinar. The title of the webinar today is C3d, Moving Laboratory Research on Pathogen Detection into Commercial Practice. So before we get started with the presentation and before I introduce our presenters, I want to point out just a few items. So first, if you haven't done so, please take a moment and answer our poll question. So we try to keep up with our audience makeup, and our goal is to really do our best to reach out to industry, and so we're trying to improve our university industry and also university government relationships. So if you'll take a moment and answer that. And next, about halfway through the presentation, we'll have about a 2-minute trivia break, and so we're going to ask you a couple of questions about the presentation material to that point. Next, at the conclusion of the presentation, we'll have the question and answer session. And so at the very bottom of the box, or at the very bottom of your screen, you should see a chat box. And so as the presentation goes forward, when your questions come up, go ahead and enter them into the chat box, and you can enter your questions in at any time, and we'll get to your questions at the conclusion of the slides presentation. And so finally, I'm going to change the screen a couple of times. And so I'm going to demonstrate that, and this will get us to the presentation slides. And so when I change the screens, it'll take just a few moments for the screen to set back up. Okay, so finally with that, I want to thank everybody again for participating. And now Dr. Mike Gladish and Dr. Eduardo Jimenez from Purdue University. Got to get the pitch in for Purdue University. So they're going to tell us more about their efforts toward the goal of providing an instrument for rapid detection of pathogens in food and water. So Mike, Eduardo.
Well, John, thank you very much, and thank you for the audience that will be listening to our work in this area. And basically what we're going to address today is food pathogen detection and concentration and recovery of microorganisms for the purpose of doing this. Just a little bit of background. I have been working at Purdue University for quite some time, and have been doing this work as part of the Center for Food Safety Engineering, where Lisa Maurer is the director of the center, and this has been supported through the USDA over a number of years. We work cooperatively with the Eastern Regional Laboratory in Philadelphia with George Paoli and his colleagues, as well as others in the industry, in order to develop methods which will improve the manner in which food pathogens are detected. With a few words, perhaps Dr. Hermanis can also introduce himself.
It's my pleasure to be here together with Dr. Letch. I'm part of Dr. Letch's team addressing some new approaches to from food. I am a bioprocess research scientist, and I also
Well, thank you very much.
Yeah, and I'll be covering more of the technical part.
Okay, and again, Eduardo and myself, Mike Gladish, would like to thank John for this opportunity to discuss our work. The first slide shows really the result of about 10 years of research. There are 2 prototypes. We're actually building a third, and we're modifying the instruments. But what these instruments do, the prototype and the replicate unit on the right, is to rapidly concentrate microorganisms from food materials directly so that they can be probed using PCR or other methods, including plating, for the purpose of detecting different types of pathogenic microorganisms against a background of naturally occurring microorganisms. So John, I cannot advance the slides. So. Oh, there it goes. Okay. So what we'd like to do today is address the problem being addressed in general terms and also the physical and economic impact on producers. We will also discuss the basic science related to the problem, and Dr. Amanis will cover this and give a little bit of background as well as the chemical and microbiological principles upon which this work is based. Next, we will move to the solution that we propose, and there are many solutions out there. So this is one of them that works very well for the types of foods that we've been working with, both meats and vegetables. We will cover what is being addressed and also the potential physical and economic benefits if this proves to be successful to the producers of food products, both meat, vegetables, and other types of foods. We will also discuss the basic science related to the solution, including the microbiology and some of the science behind obtaining successful microfiltration of complex biological fluids, which in this case are derived from stomach or homogenized foods. Last but not least, we will cover the economic benefits and discuss the status of the commercialization process and the steps that need to occur to bring this technology to the industry in order to help make our contribution to food safety. So with this, I'd just like to start and describe the fact that the CDC estimates that each year there are a large number of food poisoning cases. Not all of these are due to pathogenic microorganisms, but most are. And it is serious in that a large number of people are hospitalized, and of these, several thousand may die of foodborne diseases. And again, the websites on the presentation give the background and also the methodology for determining the impact of food pathogens and food safety. Scharf et al. Estimated the cost of the illness was measured in large numbers, perhaps billions of dollars. And again, this is determined by the methodology used, but nonetheless, it's a huge issue. And there's about 14 major pathogens in the U.S. that are responsible for food poisoning and food illness. What we will do here is focus principally on salmonella for today, just to give an example of how the system works and how rapid concentration and recovery may be used to contribute to food safety. This would occur due to the increase in the rate at which food pathogens could be detected. So anyhow, salmonella and toxoplasma are the first and second most costly foodborne pathogens in the U.S. And this is particularly true as chicken becomes more popular. And so what Dr. Amanis will do and myself later on will describe and discuss our work today on salmonella detection. So with this, Dr. Amanis will give an overview on the general approach for our project and what might be expected as we run through this particular webinar.
Thanks, Dr. Hodge. And this slide is a first overview of our approach. Initial general idea, how we and through the presentation, we'll provide more details of that. But again, as a general first overview, we start our approach sample preparation, following not only FDA but also USDA guidelines, depend on the type. And after the sample preparation, then we proceed with a pretreatment step, which again we are going to explain later with more details. And during the pretreatment steps, we make use of enzymes and membranes. And after we do this pretreatment step, we then move the next step related to the microfiltration of the processed samples. And the goal here is to concentrate cells and recover them in a level that can be used for detection of target microorganisms, either for using more common procedure or use procedures such as selective media, other methods more related to fast detection methods. Then after we collect the sample, then we start the cleaning of the system, so that not only the cleaning but the sterilization of the system. So the instrument can be used again for a second round of analysis. The following slide is to give you an idea of the objective of our, which is to develop and integrate a technology for fast and effective concentration and recovery of target microorganisms from food-related material, from food matrix, using an automated instrument, which is a continuous cell concentration instrument called C3d. So in this graph here, it's very clear that the major time-consuming step for the process of pathogen detection in contaminated food, the cultural enrichment. It can take 24 hours or more. So the goal with our approach is to reduce time for cultural enrichment, either to reduce time or eliminate, depend on the food to be analyzed and the concentration, either to eliminate or to reduce time for cultural enrichment. So once we can reduce time using a microfiltration approach, and we collect cells and recover them in a level that can be used for detection, then we can make use of not only plating for but also several different detection methods, more fast detection method available, knowledge available nowadays. Some of them approved by the, which include multi-fluid detection, and you can see in the bottom of the slide, immunoassay procedures, PCR-based procedure, bacteriophage reporter, in our case, we have been working with several of them, but with major focus on PCR-based detection. So, in summary, what are our objectives, challenges, and our approach? So we have the objective to rapidly concentrate pathogens in contaminated food. For that, our approach is to use cross-flow microfiltration. We have the objective to use an automated operation. For that, we use the approach of using microfluidic sensors and LabVIEW program software. We have the objective to reuse the membrane, aiming to reduce costs. For that, we develop sequential cleaning procedure. So after using the membrane, we can reuse that. And what would be the challenge for all of that? One major challenge is that when processing the food, fouling agents are generated, and those fouling agents, that could be carbohydrate, nucleic acid, they can clog the membrane. So in order to avoid that, it's critical to have a sample pretreatment before the microfiltration step. And the whole procedure from sample to sample, it takes in average 90 minutes, but it can take lower than that depending on the food sample we are. So here, I'll give you a little bit more detail of our procedure. So for food sampling, most often we stomach the samples. Where we first mix the food with some solution, could be buffer, in a 1:9 ratio. But in some cases, for example, for chicken carcass, following USDA procedures, generate a rinse. And once we have the stomach solution or the rinse, we then go for the second step, which, as I mentioned to you before, is a critical pretreatment step where we make use of enzymes and different types of membrane. And the enzymes are chosen based on the food matrix that we are. And the pre-filtration membranes, they have part size larger than the microfiltration membrane. Because the goal here is that the microorganisms pass the pre-filtration. So then the objective here is to have a pre-filtered solution, enzyme treated, where we eliminate most, or if not all, of the fouling agents. And we kept the microorganism in viable conditions to be concentrated and recover further using the approach for cross-flow microfiltration. So the cross-flow microfiltration, we use membranes, hollow fiber membranes, which I'll give you more, which has a part size of 0.2 micrometer. So our instrument is based on a continuous cell concentration approach. Then we call the instrument C3d. And once we concentrate and recover the cells, and as another explanation, in this case we use a 0.2 micrometer part size membrane because the goal here is do not let the microorganisms to pass the membrane. The goal here is to retain the microorganisms so we can recover them and proceed with the next step, which is the detection of the pathogen in the concentrate. So after we concentrate and recover the samples, we then can make use of more traditional methods such as I mentioned, or making use of fast detection methods or combination of different approaches. And then the system is ready to be cleaned and This slide gives you, I think, a very good idea of the type of the sample that we generate after we stomach. And in this case is hot dog. So we on the first picture, right? We can see that when we mix hot dog with buffer solution in a 1:9 ratio, we got a very challenging sample to be microfiltered. So if you try to microfilter that without any pretreatment, the membrane is going to be easily. So on the following slides, you have on the in the picture middle and in the right side, it's more to give you an idea of how homogenized hot dog looks like. The permeate solution. So as I mentioned initially, for microfiltration, we use hollow fiber modulus. We start make them in the lab. Most of the results that I will show here, it's using in-house made hollow fiber models. And the hollow fiber models that we make in our lab, they can contain 12 hollow fibers. And they are polysulfone membranes with 0.2 micrometer pore size. And the reason that we use hollow fiber membrane instead of use, for example, flat membrane, because there are some important advantages when using those hollow fiber membranes. For example, high surface area to volume ratio, flux per unit volume of membrane model, and very important, continuous operation avoids manual handling. On your right side, you have some image generated by microscopy. Cross section of the model, and you have also some pictures in the and I think Dr. Latch can.
Thank you, Eduardo. And so basically we, as Eduardo had described, originally made the modules in our lab. We now buy these commercially, which is very important as far as translating and generalizing the technology for testing in the industry, and also for obtaining regulatory approval and validation of the method. But this technology developed over a series of years, and we've built many different types of prototypes to test it. The slide shows, first of all, the original configuration in a method we called CCR, and this was basically filling a large syringe with liquid solution to be filtered, and then having a number of filters at the outlet. First of all, remove the particulates and larger material, and then concentrate the microbial cells into a small volume, manually then removing the membrane to which the cells would adhere or be concentrated, and then carrying out subsequent analyses of these cells. We then moved, and there was a lot of hands-on operation. We then moved to using hollow fibers in a dead-end configuration, and we started to automate the process, and that is the middle panel on this slide. And so in this case, the idea was to basically force the liquid into a hollow fiber membrane, use it much like we would dead-end filtration, and at the end of the process, backflush the hollow fiber microfiltration membrane to collect the material in the microfuge tube and then carry out analysis. And so this worked well, but again, there were limitations with respect to the rates and especially the volumes we could process. And again, there was a lot of manual operation. We then went next to testing of cross-flow configuration with the hollow fiber membrane, where the material would flow through the hollow membrane continuously, be collected in the beaker. And you can see the beaker, the white fluid, in the middle of the image at the upper right side of the slide, beaker. And so the fluid would be collected and continually pumped through the hollow fiber membrane. And then as the volume decreased, the cells, microbial cells present, would also be concentrated. So that started to give us some good results, and we found we didn't need, if we could appropriately use the appropriate cross flow and cut off on the hollow fiber membrane, we did not need to have a syringe pump, since pulsation would not negatively affect the operation. So we then went to cross flow with a tubing pump so that we could have a continuous operation, which is the lower left picture, and we looked at various types of food materials there. Then we went to an instrument where we automated the instrument, again with inputs from the biomedical engineering department, in particular Kurt Foster and also Jim Jones, who developed the interface, which uses LabVIEW, in order to automatically control the system. And we're now using a hollow fiber at this point. You can see it in the middle of the screen. It's that narrow straw with the orange tabs on it. And then last but not least, we then built a number. We actually built almost a half a dozen of these prototypes in various configurations to test them. And finally, we ended up building the instrument that's shown under C3d with a touchscreen monitor, control through a PC using LabVIEW, and automated operation, which included concentration, recovery, ejection of the sample into a microfuge tube, and then cleaning of the system with sterilization of all the tubings and the hollow fiber membrane so we could reuse it. And this particular instrument had different monitors, including pressure and flow rate, weight, so we could determine how quickly the material was being processed and what the flux was through the membranes. And then also for testing to make sure we had sterility and controlling the conditions so we would be able to keep the microfiltration system running smoothly. And at this point we learned, whoops, I'm not sure what happened here. At this point we learned how to control the system in order to be able to reuse the microfiltration hollow fiber membrane at least 15 times. And automated operation is critical to achieving this result. And again, the hollow fiber membrane itself is shown. It's sort of a green tag in the middle of the picture of the instrument. On the right of your PowerPoint, the two boxes are pumps, and then we also had some connecting tubing and valves in this system. So based on that, we originally did studies with E. coli, in particular GFP E. coli, and we showed that we could concentrate these in water in a short period of time. And so basically you could actually see the pellet, as you can see on the left, which was obtained from a fairly clear and dilute solution starting at the beginning. So with this, we then moved to food materials, which had a number of other challenges which we were able to overcome. And Dr. Hamanis will give us some further background on that.
So, as Dr. Ledge mentioned, we did some initial tests with cells in buffer, so we could estimate how well our system would work to concentrate and recover cells in solution. For that, as Dr. Ledge also mentioned, we use Escherichia coli. But our next challenge was to show that we could also do that using a food-related sample. And we then decided to test chicken meat extract, where we artificially spike salmonella to estimate concentration and recover using our instrument. So again, the first step was sample preparation, and we mix pieces of the chicken to in a 1:9 ratio. Then we stomach that, and the final volume of our solution was 250. And at that in this solution, we spike the salmonella cells in a, and after stomach, as I mentioned before, it would be very challenging to be able to concentrate and recover cells in this sample without to have some initial pretreatment. So after we generate the sample for stomach, we then did an enzyme hydrolysis step. And we also use, as I mentioned before, a pre-filtration membrane with pore size larger than the hollow fiber membrane, so we could remove some possible purulent agents, but also also important, recovering the microorganism in viable conditions, so that we can process then the pre-treated sample microfiltration and concentrate and recovering the cells. So in the bottom of this slide, you can see again one observation here is that in this case we use C3d one version before our latest version. So in the bottom of this slide, you can see some pictures showing microorganisms on selective medium. And in this case, as selective medium, we use the chroma agar for salmonella. And you can see that the first picture on your left is related to the original sample. So it's the sample before being microfiltered and we plate the 100 microliter of the sample with no dilution. And then in the middle, and they are representative of the concentrate and recovered samples. The difference here is how many times we dilute them. It's the same sample, just showing you a plating of the concentrate and recovered sample after 100 times dilution and after 1000 times dilution. So we were able to estimate the more than 70%, around or a little bit more than 70% recovery. And we could also, by using qPCR, also confirm the results on the detection of salmonella in this. So you can see in this table that PCR inhibitors are, especially in the case of major problem, but in this case, when doing the qPCR analysis, we could see that results for cells in buffer, when compared to results for cells in the rinse, so cells which were, results are very close to each other when you compare unspiked, artificially spiked, and very important, the whole procedure from stomach to qPCR analysis took about 8 hours. Then comes another very critical step, which we want not only to reuse the hollow fiber membrane, but we also want to make sure that the system is cleaned and sterilized for another round. So for that, we developed a sequential cleaning procedure using, first we pass sodium hydroxide through the system. But you can see in the first two pictures that sodium hydroxide cleaning is not enough to remove to kill or on the. So on your left side, again the first two pictures, you have cells sprayed on a non-selective medium. So, and before I should have said that in this case was an experiment where we used salmonella. So it was related to the previous slide where we inoculate some concentration of 10 to 4 CFU. So in the first picture, again, it's related to a non-selective medium, more specifically LB with no added to that, no antibody. So you can see that some cells survive the sodium hydroxide cleaning step. And you can see also that we also confirm this observation when we plate the same sample on agar, which again is a selective medium for salmonella. Salmonella would be what? Mauve cells, mauve color, and the blue cells are more related to coliforms. Then on the sodium hydroxide step is not enough to clean and sterilize the system. So we then pass water through the system, sterile water through the system, and we do another step, this time using ethanol for cleaning and sterilization of the system. Very critical, and you can see in the 3 pictures below that all the solutions are fresh during this step and or sterilized. Because sometimes if you see the picture in the middle and the last picture, so the picture where we plate the sample collect after we proceed this step in XLG medium, which is another medium selective for salmonella, and the chromaga. Sometimes you don't see salmonella, but if you do the plating on non-selective media, you can sometimes see 1 or 2 cells that could survive both treatment. And if you don't use, you don't pay close attention to use fresh solution and fresh and sterile solution. If you pay close attention to that, and we have been testing that for hundreds, several times, never see any contamination.
So with that, the next step would be to consider how we go through and translate the science and food safety. And just in summary, what we've shown, and we've done lots of different food materials, including ground meats, chicken carcass, vegetables, water, and other types of processed foods. We have gradually improved the processing, so we're decreasing the time required from start to finish. Our goal being at least for now, 6 hours or more quickly. This includes the detection part. As Dr. Hermanis had mentioned, qPCR is really a major factor in rapid detection. There are some other methods that are now recently developed, and we are discussing these with some of the suppliers. So the research is continuing. There's lots of different elements that are brought together, but we believe by having the prototype, which you saw in the very first slide and was repeated several times, we're able to package the science, which by the way has been published in a series of papers in Applied Environmental Microbiology, in AI Chemie Journal, in Biotechnology, Bioengineering, and other journals in this area. And I'm sure John could provide you a list of publications. But what we've done is packaged, hopefully, our knowledge in a form that others can use. And so really it's a matter of engineering the science and applying this to food safety. And so we looked at the molecular and microfluidic phenomena of microbial hazards in the food supply. Some of the other areas we have looked at and continuing to study, for example, is to what extent microorganisms adhere to different foods. And there are differences among different foods. And what you'd like to do, of course, is be able to get the microorganisms in a living form, away from the food, in a liquid extract, so you can then further probe for presence of pathogens. We've been able to translate the fundamentals into instruments, and also we've begun to apply process control in the instrument with the idea that if ultimately you could have hands-on, hands-off operation, the results from the instrument coupled with a rapid detection method on board might serve as a first alert in a food manufacturing facility, although this still needs to be fully developed. The science that we've looked at is molecular biology and the biochemistry of the biological agents and of the organisms that we're studying. The threshold levels needed to be detected in the food system, which, by the way, we have found regardless of method, there has to be at least 1,000 microorganisms in the sample presented in the method to detect one pathogenic organism. We've shown this in a number of ways. We've also looked at a molecular level the binding of biomarkers to pathogens. We've developed membranes previously which actually selectively capture certain pathogens based on polyclonal antibodies that we've developed with some of our colleagues in food science, including Arun Bunia. So basically that brings us to food safety and the consumer. And basically salmonella is a big issue. As you can see from some examples, that some of these are dated, they're more recent ones than these, but these are from the Senate, US Senate, and basically just shows you that there are issues with salmonella, E. coli, and less so with Listeria monocytogenes, but we have studied all three of these. There's other types of pathogens that may be found in water. We're also doing research in this where we concentrate large volumes of water to probe for these. But basically, I think there's an issue there, and I think rapid detection will be one part of an overall process which will enable implementation of methods and manufacturing protocols that will minimize the risk of pathogens being found in food that reaches the consumer. And the detection and the prevention really have common elements, and that includes the solid and liquid distribution of microorganisms in the food versus what you find in the liquid. The surface in the liquids and the surfaces can be membranes, absorbents, beads, and there are certain methodologies, for example, that use derivatized magnetic beads. But again, having a high enough concentration is critical to the success of the detection methods. What we try to address here is improving the detection method by shortening the time required for bringing the microbial cultures to a concentration or a number that the pathogen can be detected. And so we need to obtain a large representative microbial population to interrogate for the presence of pathogens. And after that, we're looking at detection by biomarkers, PCR, or plating methods. We always do the plating anyhow because this gives us a nice confirmation of results that we get much more quickly using some of these other methods. A key is maintaining the viability of the microbial population throughout the concentration, recovery, and detection steps. So the current food safety challenges is that food safety will continue to arise in unpredictable ways. And if you follow some of the recent incidences, that pretty much, unfortunately, is still the case. And it also will reflect changes in food production and supply, changes in the environmental and the environment leading to food contamination, a rising number of multi-state outbreaks, which means you'd like to catch a problem before it is distributed to the consumer, as well as new and emerging toxins, antibiotic resistance, and microorganisms, and so forth. So these are all areas that need to be addressed and will require eventually rapid detection methods, as well as, more importantly, methods in the production and the supply chain that will minimize the risk of contamination. So really what one needs in order to bring a technology to the marketplace is a clear vision of what the business model might look like. And this is a slide, or this slide is not directly from Hamermesh, but basically is from a Harvard Business School case study. And Hamermesh does a very nice job of describing what needs to be done in order to develop a business model, and also defines it very clearly. And that's really a summation of core business decisions and trade-offs that are employed by a company to earn a profit. Basically how a new venture is going to make money. And so although we as scientists and engineers are really interested in the technology and of course disseminating and sharing our knowledge with others in the field so we can continue to improve food safety and methods of detection, we still need to find ways of bringing this technology and packaging it in a manner that the food industry could use and in a manner which it can be validated using existing organizations that the food industry works with. And to do this requires capital, it requires ongoing businesses to handle this methodology. And assuming this were to work out, one could imagine that we would need to have hundreds, if not thousands of these instruments. And although the membranes now are commercially available, which is a wonderful development, the instruments themselves, we've made, I think, over the years, probably about 10 or 12 of them. We're not really in the process of manufacturing instruments. However, if we can come up with a business model that would make it worth the while of some of our potential collaborators, and we're looking into this, that would then help generalize and bring this technology to the industry and hopefully improve food safety. So anyhow, as we get to the last few slides, the business decisions and trade-offs that we've begun to consider include revenue sources. Where would they come from? The type, would they be through sales, service fees, advertising, etc., and the importance and relative size of each so we can segment the market and approach the appropriate company or companies to help us commercialize this technology. I personally have also started up a company which was acquired in a very different area and have had some experience in this. And so I'm bringing this general experience together with some of my colleagues at Purdue. They have a very active program in translation and commercialization to try to bring this technology to the industry. Although it is a very significant task to do so. And the other things we're looking at is cost drivers. Cost is going to be critical in this application. The costs must be measured probably, we're guessing, in the range of dollars per sample for this to even be considered by the industry because of the large number of samples that ultimately would have to be tested. And as we all know, there's a large and viable segment already which does testing of food products for the presence of bacteria, for the presence of pathogens, and so forth. Although most of these are culture-based methods or enrichment and selective culture-based methods coupled with PCR, in some cases immunoisotope type assays. Nonetheless, most of these methods require, or all of them that I'm aware of, require more than 8 hours to be done. And so this is where we're trying to couple with existing technologies some methods that shorten the time of enrichment and give us larger populations of cells to work with for the purposes of interrogating them and for the purposes of detecting quickly pathogens if they're present. So the investment size is another area we're working on to estimate how much it would cost to bring these first units, and we are working with some colleagues on this in the industry, as well as critical success factors. And again, these are right out of Hamermesh case studies, and we're following these, and we find that there are many elements past the research, past the prototype stage, that must be addressed in order to achieve successful translation. So the business model generation requires all these questions, and this is a chart that we're looking at and developing further, and this is sort of a more detailed version of this chart. And basically, we're going through each of these questions in order to basically come up with a 2-page summary of what needs to be done next. And by being done next, I'm thinking in the next 6 to 9 months in order to take this to the next step, even as the research continues and even as the technology is improved. So the question is, on a J-curve, basically what happens is there would be significant expenditures as this technology moves from our laboratories and from the USDA into commercialization and making this more widely available. And so really what you can see is the major steps, and these are called the 7 factors. This is a methodology that I've developed together with Professor Natalie Duval-Coutil at Purdue University, where we teach this in a technical entrepreneurship class. And so anyhow, it's discovery, disclosure, which we've done, the development, which you've heard about, and the prototype. And we're really at step 3, and we've started market assessment. So there's all these other steps that still need to be done, and some of these may have off-ramps, but they will require more capital. And so this is a very important component of bringing a technology such as this into an industry in a form that may be quickly validated and hopefully contribute to food safety. So with this, I'd like to end our presentation simply to say that food safety and the foodborne pathogen detection is very important, is becoming more important. And we hope that we can, in our own way, at Purdue University and through our funding from the USDA and other sources, be able to contribute to solutions of a very complex area. So with that, I'd like to thank you very much, and would be open for questions, realizing, of course, that we still had the trivia questions to address, which we passed over accidentally, but we can answer those questions as well as we go forward. Thank you. So the trivia questions really were, and Eduardo, over to you.
Yeah, the first question was, what was the major
Well, here I'm
advantage?
going to go. I'm going to go ahead and pop these trivia questions up real quick. So let's give the audience a second to answer these. Sorry, we did get off format a little bit, but we'll come to that. So go ahead and take a look and answer the trivia questions. And we'll take a couple of minutes for that. Also, while you're answering the trivia questions, if you wouldn't mind, go ahead and put in your questions in the chat box below. If you click on everyone and then type your question and then hit enter, you can get those questions in. And it looks like we've got a couple that are popped in here. So we'll get to those in just a moment. So we'll give everyone here just a couple of seconds to get their thoughts together about the questions. Okay, well, I'm gonna just a couple more minutes. I'm going to change the screens real quick so we can see the chat box a little bit differently. And I'm gonna move the questions off the screen. Let me change the screens. Put these trivia questions back here for just another moment. Let's move this one down here. So we're going to improvise our format here just a little bit. Okay, so gentlemen, I'm going to go ahead and ask this first question. So it comes from Ruth, and she asks, how does this filtration technology compare with the Innova Prep?
We are familiar with the Innova Prep system, and that is a straight-through system. So for those of you that have not seen it, if you go to their website, they have a really nice picture of it. But basically, this Innova Prep system, as far as we can tell, is effective and is similar to some of our earlier work where you had flow-through method. In the cases where solutions are clean, the Innova Prep works quickly, and I would guess that if you address questions to them, they could, more in detail answer some of the other technical features of their system. We have not used it, however, so I do not know more about it, but it's something that we have seen and actually would like to try as well to see how it works relative to what we're doing and to see if there are ways to combine the two methodologies to get even a better result.
Okay, thank you for that. So the next question: have you worked with? I'm gonna butcher the pronunciation. I'm gonna bet here, but Legionella detection.
No, we have not.
Okay. So while we're again, I think just a little bit more about your questions, and go ahead and type those in. I've got a couple for you. This is more related to the kind of the tech transfer side. As an ag economist, I'm interested in the entrepreneurship part. So on the commercialization side, can you just talk real briefly about your experience working with the Purdue Tech Transfer Office? What has that been like for you?
Well, we found the Purdue Tech Transfer Office and just Purdue University in general, at least over my experience since about, maybe going back about 10 years, has been really positive. And basically, we're able to translate the technology. They're encouraging university professors such as ourselves to be involved in startups. The university side has made it possible. In my case, I took a leave of absence. This is in an unrelated business, a partial leave of absence to in fact, translate and transfer the technology. And they've been very positive also in developing facilities in the Purdue University Research Park, which I think has probably several hundred startup businesses of various types. So in general, it's been quite positive.
Okay, well, it looks like we've got a couple of folks that are typing questions right now, so hopefully we'll get those popped up here in just a second. Okay, well, let's go ahead and get to the next question. So what about the accuracy and the cost related to your technology?
Okay, the accuracy, I'm not sure what that question pertains to. It is reproducible. If the accuracy refers to the detection part, that's really a function of the sample that is presented to, let's say you had a PCR instrument, let's say you had an immunoassay, assay. It really depends on the number of cells in the sample, and in the case of PCR, whether or not inhibitors are present. In our case, as you can imagine, because of the way the concentration microfiltration is doing, inhibitors, as far as we can tell, PCR inhibitors, as far as we can tell, are removed. So really, if we can get to about 1,000 viable cells, regardless of the methods, the detection part relative to plating methods, which would take a day, but the detection part that's done with PCR or other rapid methods seems to be consistent with the plating methods. As far as the cost goes right now, we have not paid a lot of attention to the cost. We have, but we haven't. In other words, we don't have a cost analysis which is suitable for a business, but we do know that we have to get down into the dollar range or several dollar range per sample, not higher than that to be practical. And so that then really depends mainly on the number of times that the membranes can be reused and the amount of solvent that is required to go through each cycle. And that's being optimized at this point.
Okay, thank you for that. So our next question, you said the hollow fiber membrane could be used about 15 times. Can the same hollow filtration tube be used for concentration of more than one organism sequentially? For example, concentration of salmonella from chicken rinse, cleansed, then concentration of E. coli from water.
The answer is yes, although we have not done this sequence that is mentioned, salmonella and E. coli. But the answer is yes.
Okay, thank you for that. Well, so I've got one last question for you, and if we don't have any questions after that, then we'll go ahead and wrap up here, and I'll pop these trivia questions up. We can take a look at that. But so from the again, thinking about it from an economist perspective, from the entrepreneur side, what are some of the other considerations you're currently making in the technology development? So I kind of go back on the cost side question, but what's kind of the next step for you from the entrepreneurial perspective? If you could elaborate on that just.
Well, from that perspective, I think the key is to work with others, with the industry, in order to validate this methodology, make sure it meets the requirements of the USDA or FDA, depending on the type of food that's being tested. We're also working with our colleague at Purdue University, Amanda Deering, and she is quite an expert in the vegetable area. And so again, in working with her, we have learned that the requirements, or the way things would be tested for vegetables, for example, not surprisingly, is different than ready-to-eat meats, which would be different from fresh meat. And so the main thing is, the next step is to demonstrate this instrument in a way that we can begin to validate, let's say through AOAC, its operation. And it basically, again, perhaps pertains to Muhammad's question on what about accuracy, to show that if we have this instrument working properly, we're able to minimize the number of false negatives, certainly, and hopefully false positives. But to get this statistically, you'd have to run a lot more samples than we have. We've run perhaps hundreds of samples, or maybe thousands. I don't know, a large number of samples, but nowhere near enough, and nowhere broadly enough to be able to validate the instrument for all different types of foods. And so that's really the next step. To do this practically, one needs to have close collaborations with industry, as well as the USDA and the FDA.
Okay, thank you for that. Eduardo, do you want to share the results of the questions, the trivia questions,
Yeah,
what the actual answers were supposed to be?
Yeah, the first question was what is the major advantage of using a microfiltration approach compared to a standard method? You can see that everybody answered the reduce time for pathogen detection, eliminating or reduce time for. And the second question was, what is the most significant challenge for? And again, those who answered, the answer minimize falling effect of food particles.
John, I noticed that many people have said all of the above, and we appreciate the compliment. Practically speaking, we think that yes, it can be coupled to different methods, and it is, I don't know about the reliability of the standard methods, but the single major advantage we believe is the reduction in time. Similarly, in the second question, The most significant challenge is fouling effects, but certainly all of the above also would pertain in that we want to be able to couple this instrument with a recovered sample, and then also basically choose the appropriate detection methods, although we've actually started working on C with some companies in order to basically show that this methodology may be beneficial in complementing the performance of existing instruments, which, by the way, work very well already.
I think the classic all of the above answer sometimes is a trick question or a trick answer.
Well, it's
Okay, well,
unfortunate part about academics. Right?
Oh, yeah. Yeah. Okay, well, I've changed the screen, so for those of you that are still with us, I've got a couple of other questions related to tech transfer. If you would not mind taking a moment and just responding to those, and so I want to thank everybody for your participation. Mike, Eduardo, special thanks to you. And so I've got a couple of web links up. So I've got my email address, and I've got email addresses for our two presenters. I've also got my LinkedIn account up. So I'm a big fan, a big advocate of using LinkedIn for making industry network connections. So please connect with me there. And if you'd like a copy of this recording, please send me an email, and I will email you a link. My mailbox, I've got about 30 or 40 requests for a copy of the for the webinar, since there's several folks who indicated to me they weren't able to participate. But again, gentlemen, thank you for the presentation. It was very informative, and I appreciate your time. Any parting thoughts?
Well, we thank you very much, John, for this opportunity, and we hope this was useful to the audience. Dr. Amanos and I also very much appreciate the support we have received from the USDA and from other sources of funding, as well as the work of our colleagues through the Center of Food Safety and Engineering, which enabled us to do all this. As you can see, there is still going on a lot of fundamental research, but also a lot of effort in developing the prototypes. And we do very much appreciate the support we have received to do this.
Okay, well, with that, we will wrap up this webinar. So thank you again.
Thank you.